Gel electrophoresis is a very important technique when we start to talk about biotechnology, because this is going to enable us to separate a variety of molecules from each other. And by definition, this process of gel electrophoresis is separation of molecules based on their movement through a matrix in response to an electrical current. So the matrix part right here, that's where the gel comes into play. And the gel is kind of like a jello type substance. It will have very small gaps within it. So you'll have a lot of molecules that are closely gapped together. And this is going to create kind of a obstacle course, if you will, for molecules to move through. So if you have a particular molecule, it's going to take a while to navigate its way through this gel. So the purple is representing the molecules. Now the current part of it, the electrical current, that's the electrophoresis part of it, we will have electrodes that represent various charges on the two sides of this gel. We might have a negative electrode up here and we might have a positive electrode up here. And then if we have a charged molecule itself, it's going to want to move in response to the electrical current that's being applied. And so remember that opposite charges attract. So based on that principle, it's going to be very useful for molecules that have charges, because they will want to move through this electrical field that we're going to be applying through this process of gel electrophoresis. Okay, so the two keys here is that we have a matrix, which is our gel, which will have a series of pores of a specific size that the molecules are trying to navigate through. And then we'll also have an electrical current that is going to kind of make them want to move through that obstacle course of the gel. So here we have another representation of this. So this square piece that you're seeing here, that's the actual gel that we're going to be having our molecules move through. And then we have within that gel, we have a series of holes which we call wells. And so these are really where we're going to actually load our samples. So we would put a solution that contains our molecules into those wells. And so I'm going to draw that here. And so we have a whole bunch of molecules in there. And then once we load them in there, we cover it with a buffer that's going to be able to conduct these charges. And then once that electrical current is applied, the molecules are going to want to move. So they will start to move towards the opposite charge. So if these are molecules that tend to have a negative charge, they're going to want to move towards the positive charge. And so if we think about DNA, DNA is overall negatively charged because of the phosphate groups that are present there. So DNA tends to move towards the positive. And besides the fact that it's attracted to the positive, it's also repelled by the negative, because like charges repel each other. So what happens is that these molecules start to move through the gel, and based on their sizes, some of them will make it through the gel faster than others. So we'll start to see spreading out. So if we were to draw these molecules, you might have some that make it this far, and we have different distances that they've made it through the gel based on their particular size. And as it turns out, smaller molecules move faster. And so if the smaller molecules move faster, these towards the bottom, these would be your smaller ones. And then the slow ones up here, those would be the larger fragments. And so really what we're doing here is just overall size separation of the DNA. We can alter the percentage of the gel matrix, so we can make the pores of the gel a little bit tinier, or we can make them a little larger, which does affect the overall rate of movement. But no matter what, the small molecules are going to be the ones that are able to just navigate through these tiny little holes faster than the larger ones. So this is a very useful technique if we need to separate molecules from each other, specifically nucleic acids like DNA and RNA. This is used very commonly for them.